Thanks for that nice introduction. Harvard's renowned for these incredibly long titles, and so I just find them quite embarrassing. So first I wanted to start by saying I flew in last night from London, so everything's still a little bit hazy, and it's hot here. So I'll do my best to be as coherent as I can. I welcome any questions. If you guys have questions, please interrupt me, and we can make this as informal and as fun as we possibly can. So today what I wanted to do was give you an overview of one of the big projects in our group, because I think this actually makes for a very nice example of natural selection and sort of has all the components together. So I've worked with a number of high school teachers trying to modify this as an example that folks can use in the classroom as a very intuitive and compelling example of natural selection in the wild. And so I would like as much feedback from you as well in how to convey this and how to make this accessible to folks that are looking for examples of sort of evolution in action. So I like to start by talking about Darwin, and this is in part because last year, of course, we celebrated Darwin's birthday and the anniversary of the publication of The Origin, so there were countless festivals and seminars across the country, but it really was nice because it sort of put everything in perspective, because a lot of us went back and sort of revisited Darwin, went back and read The Origin, and so forth. And so I always feel remiss in sort of starting a talk about evolution without at least mentioning Darwin, but instead of talking about what Darwin knew and everything in all his prescience, what I'd like to do is talk about what Darwin didn't know, and that's the genetic basis of adaptation. So we have to remind ourselves that at Darwin's time, we didn't know anything about genetics, the mechanism of heritability, which makes his discoveries even more sort of impressive. So I wanted to start by just sharing a little anecdote about Darwin that was discovered in 2004 by Matt Ridley, who's a science writer, that inexorably links Darwin to the second great discovery, that is the discovery of DNA. So what you're looking at here, and I of course don't expect you to read this, but just sort of to be impressed by it, this is Darwin's last publication. It was published just 2 weeks before his death, and it was published in a prestigious journal called Nature. The title of it is On the Dispersal of Freshwater Bivalves. And why this got published in Nature is a story I can tell you about later. But the main point of this note was to report an observation of a little water beetle, and clamped to the leg of a water beetle was a little clam. And this explained how clams could migrate from lake to lake. And that was the importance of the paper. But the thing that I wanted to mention today is sort of how Darwin got his hands on this little water beetle with a clam on that. And it was sent to him by a young shoemaker in the British Midlands, who was also a naturalist. And his name was Walter Drawbridge Crick. So that should ring a bell, because almost a century after the publication of The Origin, that shoemaker's grandson and his friend Jim Watson made that second great discovery, that is the three-dimensional structure of DNA, which of course filled in that black box that Darwin didn't know about, and that is how traits are passed on from generation to generation. And this is really sort of where our work comes in, because it's in this text that we find more evidence for Darwin's theory of evolution by natural selection. We find evidence for our 3 billion year existence, the shared common ancestry of all living organisms, and then what I want to talk to you about today, and that is the mechanistic details about how this diversity evolved. So the precise DNA base pair changes that are responsible for the diversity that we see in nature. And so that's what my group is really interested in, and that is sort of this big question of how this evolved. And the main question I want to talk to you about today is this: what is the genetic basis of fitness-related traits? And what I mean by this is, what are the precise DNA base pair changes that give rise to variation that matter to organisms in the wild, that help them to survive and reproduce? And this is sort of thought of as the holy grail of evolutionary biology, or at least the part of evolutionary biology I'm working in. So today what I want to do is sort of try to make these links for you. That is the link between environment and phenotype and genotype. Then think about this as two different levels, sort of the ultimate level at which phenotypic variation, the outward appearance of organisms, why they vary, and how that's connected to the environment, so changes in the environment that may promote variation in phenotype. And then I want to take a step sort of more at the proximate level and try to understand the genetic changes that are responsible for this. And I think when we make these links, we talk about things like natural selection, so how natural selection promotes phenotypic variation. And then we bring in these more molecular sides of the story, that is understanding the genes that underlie phenotypic variation that's important for fitness, but also how changes in these genes through development actually produce that variation. And then I think this is when things get really interesting, because once we have all the components, all the pieces to this puzzle, then we sort of have a bigger picture of adaptation. We can go back into the wild and ask how these traits evolved. So there are lots of ways to sort of approach this question of how to link genotype and phenotype and environment. And the way our group has been approaching this is to start with a phenotype that we think has some effect on the way that organisms, in this case, the case I'll talk to you about is how they survive in the wild. So the phenotype I want to tell you about today is color. And that's because color is one of the primary ways in which organisms interact with their environment. It's involved in a number of biological processes. So here are just some examples. It's involved in mate choice, which of course is associated with reproductive effects. It can be involved in warning coloration, similarly in mimicry, and what I'll talk to you about today is crypsis. So organisms camouflaged in their environment to decrease the amount of predation and increase their survival. So color is fascinating not only because it's intuitive, it's easy to measure, we often know, at least somewhat what its role is in nature, but we can also, if we're interested in the genes underlying color variation, we can also take advantage of the fact that color has been studied for over 100 years by geneticists. So what would happen is you'd have these lab colonies of mice, and every once in a while you'd have this mutant mouse that would pop up, mutant in the sense that it was a different color than the other ones. And then geneticists could do crosses, try to find regions of the genome that were associated with these color differences, and then ultimately find the genes. And of course, this has been in much more recent times that we've actually been able to find the genes, sequence them, and find the mutations in these genes that are responsible for the color variation. So again, I don't expect you to read this, but just sort of to appreciate that we know over 100 genes that are involved in coloration. In other words, if you knock out any one of these genes, you're going to have an effect that's visible to a geneticist or a grad student who's taking care of a lab colony. And not only do we know what the effects of mutations in these genes are, but often we know if there are other effects, that mutations in these genes cause other effects other than just on color. So here's a classic example. This big yellow mouse here is a mutation in a gene called Agouti. It's called the obese yellow phenotype because the mice are not only yellow, but they're also obese. That is, they keep refeeding and refeeding and refeeding until they get about twice the size of their other counterparts. And so that's also important information if we want to know, because in the wild that could actually be very deleterious if you just have to eat all the time and you're out foraging all the time. So this information is actually very useful to us, even if we're not interested in lab populations of mice and we're really interested in wild mice. So unfortunately, these lab mice, even in the wild, so they're house mice. These are the types of mice you would catch in your house, unless you live in the country. But usually in the city, you'll catch these kinds of mice. They actually, because they live in houses, don't vary a lot in color. So we've been studying a group of rodents that are closely related to house mice, but they in fact do vary in color. And I warn you, this next slide. This is actually how we prepare mice. So these are old museum specimens. They're housed these guys are housed at the University of California, Berkeley Museum, but we have specimens that are maintained like this. And it's actually a great way to prepare mice because you can actually see the color variation very well. Okay, so this is a group of mice called Peromyscus. They're deer mice. If you live out in the country out here, these are probably what you catch in and around your property. And what's great about these mice is they vary a lot in color. And they vary in color in two different ways. They vary in the overall coloration of their dorsal coat, so the back of their coat, ranging from white to almost black. But they also vary in the distribution of pigment or pigmentation pattern. And so what's fun about this, working with these mice, is that a lot of the genes I showed you on the previous slide are responsible for this kind of coloration. So the spontaneous mutations that arise in laboratory colonies tend to just change the overall coloration of mice. So we actually don't know very much about how patterns are formed. So, I mean, this is a relatively simple pattern, but how tigers get their stripes or leopards get their spots. So this, working on these mice offers an opportunity to maybe learn something new about patterning as well. So these mice, in addition to varying in color, also have been the subject of study for over 100 years. Again, by this fellow. This is Francis Sumner. He sort of started it all off. This is him in his field gear, so when we go out in the field, we don't dress quite this nicely. Looking very dapper. But he was a natural historian that really spent the bulk of his career driving around North America, just catching mice and describing variation in their phenotype. Primarily, he focused on morphological change and was really fond of color, and we'll talk more about that in a minute. But he also described some behavioral variation as well, and we're working on some of the projects. So a lot of actually the work that's done in our lab really dates back to Sumner's early papers in the 20s, 30s, and 40s, because he was the one that was out there describing the variation. And now we have the molecular tools to go out and actually try to understand the genetic basis of that variation. So arguably, Sumner's favorite species, and this is mine too, is this particular species, Peromyscus polyanotus. It's called the old field mouse because it occupies old field habitats that look like this. They're sort of overgrown agricultural fields throughout most of the southeast, sorry. And so you can see Georgia, Alabama, South Carolina, and northern Florida, so that gray region. They look probably like a typical mouse. They've got dark brown coat, a light gray belly, and a striped tail. But what's particularly interesting to us about these mice is they've also recently colonized the barrier islands off the Gulf Coast of Florida and the Atlantic coast of Florida. So each one of these numbers represents a different subspecies of what I'll refer to as beach mice, because they actually occupy the beaches. And I'm going to start by focusing on one of these populations, this one, which is Santa Rosa Island. So these mice that have colonized these beaches live in habitat that's really different. You can see just by looking at this picture, it differs in two sort of fundamental ways. First, you can see the sand, in this case, or substrate color is much different. So I don't know how many of you have been out to the Gulf Coast. It's beautiful. It has the nickname of the Redneck Riviera. And if you've been there, you know why. But the beaches are actually quite beautiful. It's almost like walking on sand, or on sugar. The sand is so sort of fine and white, as compared to the dark loamy soils of inland Florida. And then the second thing is that there's much less vegetative cover. So the primary vegetation out here is sea oats, which are also the food for these beach mice. But in these areas, they experience really high levels of predation. And their primary predators are thought to be owls, herons, and hawks, as well as some visually hunting mammalian predators like coyotes and foxes. And I'll tell you more about those in a minute. So then it's maybe not surprising that the mice that occupy these beaches actually look quite different. So I should mention this is not to scale, so these guys are only about the size of a ping pong ball, both the mainland and beach forms.
You said a ping pong ball?
Yeah, about the size of a ping pong ball. So they're really quite small. And actually, if you hold them, they're actually quite docile. They'll sort of sit in your hand, at least for a few seconds, like this. So they are really quite cute. But you can see just from this picture that there's a lot of difference in their coloration and their color patterns. So these guys lack pigment on their nose, their flanks, and if you could see the tail also doesn't have a stripe. The other thing I should mention is that we know something about the age of these barrier islands. They're only about 6,000 years old, so this color difference probably arose quite recently. So it's intuitive then that being a little light-colored whitish mouse running around on these beautiful white sand beaches would be advantageous, right? Because they blend into their environment. But of course, what we wanted to know is we actually wanted to document that this was beneficial. So we wanted to do an experiment. We wanted to know how beneficial it was. We wanted to estimate the strength of the selective advantage. And then we wanted to know what was causing the selection. So first what I want to do is tell you about this link between environment and phenotype. And that is to try to understand exactly what the selective pressure, the strength of that selective pressure is in shaping this phenotypic variation. So the ultimate experiment, if we could do this experiment, it would be great. We could take 100 light mice and 100 dark mice and release them both in dark habitat and light habitat, maybe tag them and then come back maybe a month or two later and see who survives. And we would predict dark mice would survive better in dark habitat and light mice would survive better in light habitat. Right? So unfortunately we can't do that for a number of reasons. So we did sort of the next best thing, and that is we made mice. So this is my postdoc, Sasha Vinieri. She made hundreds of these little plasticine mice. Actually, we made a mold of not alive, but a dead mouse. And we filled this mold with this plasticine it's harder than Play-Doh, but if you can leave impressions in it, and that'll become important in a minute. And then she painted half of them dark to mimic the dark mice in the mainland, and half of them light to mimic the beach mice. And then what she was able to do was essentially go and march along transects, leaving mice along the transects, and then come back the next day and see which ones were attacked. Okay, so this experiment is, of course isn't with live mice, but in some ways this is could be thought of as a better experiment than using live mice, because these live mice could differ in other traits besides color. For example, the beach ones, just by handling them, you know they're much more docile than the mainland ones. And so there could be all sorts of other behavioral differences. They could have different odors, et cetera, different escape behaviors. Here, the only thing the only difference between these guys is color. But the downside of this experiment, of course, is we didn't know if it would work. Were we going to be able to fool predators into attacking these little mice? Well, we got lucky, I think, in the sense that it did work. So here's a dark mouse on light soil. And so here's its head, and the end of its tail is up there. And you might be able to see it sort of missing a chunk of its ear and a chunk out of its back. So this is a predation event. And we can tell it was an avian predator because there's a bill imprint left on its back, and there's no footprint surrounding the mouse. So what Sasha was able to do, she worked with a couple of undergraduates. She was able to mark predation events in both the light and dark habitat. And so we went out to sites where we knew light-colored mice live and dark-colored mice live, and we knew that we were surveying their natural predators. And so here are the results of that experiment. So if you just look at the top panel, those are our cryptic mice, so light mice on their light soil and dark mice on their dark soil. And then here are the non-cryptic or mismatched mice. And then what these bars are telling you is the proportion of predation. And the two important points are that in the light habitat, there was much higher predation on the mismatched mice than on the matched ones. And then the second important point is that it looks to be symmetrical. That is, these guys survived as well as these guys did. So when you're dark in a dark habitat, it's just as beneficial as being light in a light habitat. And it's just as bad in both cases to be mismatched. We can also use this type of data to estimate the strength of selection. And I won't go into details, but other than to say that it matters a lot. You have about a 50% increase in your probability of survival, that is not being attacked, if you match your habitat. So this is a pretty powerful selective agent. And then we could also tell what the sort of agent of selection was, the predators. We had marks certainly from hawks and herons, and then evidence from coyotes and foxes, and it was about 50-50 the hit rates. And then we had a bunch that we couldn't quite tell for sure, but based on the ones that we could estimate. Okay, so I've hoped to convince you so far that color matters for survival in these mice, and that natural selection is driving these color differences. So next what I want to do is tell you about the genetics. So how do you go from having mice that are light and dark to actually finding the genes that are responsible for these changes? So again, I just want to give you an overview of the type of approach that we used. We used what's called a forward genetic approach. So we brought mice, dark and light mice, in from the wild into the lab. And then we could do crosses. So you'd put a dark one and a light one in a cage together, look at their hybrid offspring. And I'll show you a picture of that in a second. But then you can take, so each one of those hybrid offspring are going to have one dark chromosome and one light chromosome from each of the parents, right? And then you take those hybrids and you cross them together, and then what happens is that shuffles up their genomes, and that's what's sort of represented here. So each individual of that second generation hybrids are going to have different combinations of alleles from either one of their parents. Okay? So we're going to see a lot of variation in that second generation, because some are going to have more pigmentation genes from the dark parents, and some are going to have more from the light parents. And then what we do simply is use a whole bunch of markers throughout the genome that tell us which parts of the genome are from that parent versus that parent. And then we measure their coat color phenotype and simply ask, is there a statistical association with a particular region of the genome and their color? So do all the mice that have completely light tails have a similar region of the genome from the light parent? And that allows us to then, sort of with this statistical package, try to identify regions of the genome that are statistically associated. And then we go back to that wonderful list of candidate genes that I showed you in the beginning and ask, are there any candidate genes in that region? Now, at the time we did this study, we didn't have the whole genome sequence from Peromyscus, but the gene order is largely the same with both lab mice and lab rats. And so we could just look at their genomes, the same region. And then the hope was to find a gene that had a mutation in it that contributed to the differences in the parents. So let me walk you through this a little bit. Okay, sorry, here are mice again in their flattened form. So here's our light parents, our dark parents, and here's what the hybrid looks like. Now again, the hybrids have one chromosome copy from each of their parents, so they're all heterozygous throughout the genome. And what you can see is that there are some characteristics in these F1s that are similar to the dark parents and some similar to the light parents. So for example, none of the F1s have a striped tail. So these guys have shortened tails only because we were taking DNA from the tail tip, so they're a little bit shorter than normal. But they don't have a striped tail. The F1s don't like this parent, okay, which suggests that the light genes are dominant. Light alleles are dominant for this parent. But they have fully pigmented faces like this parent. And so there are some genes in terms of adaptation that are recessive. Okay, so just by looking at what the F1 looks like, we can already say something about dominance and recessivity. Okay, then I told you we take these F1s and we cross them together, and now we're getting shuffling of the genome. And that's where we get a lot of variation among these F2s, because they have different combinations. Some have, let's say pigmentation gene 1, so these guys probably have more alleles from this parent, whereas these guys have more alleles from this parent. Okay? So then we measure the color phenotype of all of these guys, and we have markers throughout the genome, because this is where we're going to do our statistical association. And so just to run you through this, so essentially these are the chromosomes of Peromyscus polynotus in order of, mostly in order of size. And then each one of these markers represents a different SNP in the genome, some sort of marker that tells us whether a particular F2 has an allele from a dark parent or a light parent. And this allows us to do that statistical association. And what we found was that there were 3 regions of the genome that were statistically associated with the color differences between the parents. And in all 3 regions of the genome, we could go back to that list of candidate genes, and we found a gene. And sometimes I put these little lightning bolts in the corner of slides because this is one of those slides that represents about 3 years of work all in just a single slide. And so it's just a nice reminder that even though this is just a single slide, this is actually a lot of work by my postdoc, Cynthia Steiner. Anyway, so what I'd like to do is tell you about one of these genes, the melanocortin-1 receptor, and then summarize a little bit about the other two. Okay. So this is we actually know a lot about the structure and function of the melanocortin-1 receptor, in part because it's been studied, especially its role in color variation has been studied mostly in domestic animals and in humans for a number of years. So it's a G protein coupled receptor, and it has this typical structure of having 7 transmembrane domains. It's got these extracellular and then intracellular loops. And I've marked each one of these circles represents an amino acid, and I've color-coded them. So mutations at this one have been known to cause darkening color, and this one to cause a lightening color. So the main points I want to get across with this slide is that we know a lot about the structure and function, that we know that even single amino acid changes in a number of species can cause a change in color. But when we sequence this gene in mainland and beach mice, we didn't find any of the changes that have been previously described. But we did find one difference between a mainland and a beach mouse in the entire sequence. Only one nucleotide was different. And that caused a change in amino acid from an arginine to a cysteine at position 65 that fell right here in the receptor. So again, it doesn't overlap with any of the known mutations, but it's a new mutation.
So the other amino acid changes that are on there, those are all the other examples you showed, that all the different other documented genes that are in charge of.
So all of these represent cases in which an amino acid change has been associated with a color change in another species. And so, for example, golden labs and black labs are caused by a mutation in this gene. And I'll give you some more examples of this. In humans, there's an allele of MC1R that's at very high frequency in Irish populations. If you have really red hair and really pale skin, then you usually have 2 copies of this receptor, which is sort of a weaker receptor. It doesn't signal as well.
But between the beach mice and the field mice,
this is
that
the
population difference. Everything else is the same. So the other stuff is just other types.
Yeah, this is just other stuff in all sorts of other organisms. Yeah, and so it would have been great. The reason I put this up there, it would have been great if it was associated with a mutation that already had been documented in another species, because then we know that it was functional. But here we're sort of stuck saying, okay, here's this mutation. All the beach mice have a cysteine, all the mainland mice have this, but is it really doing something? And so here's where we want to actually test that mutation has an effect. And we can actually do these very nice, simple pharmacological assays to test this. So let me first tell you about what MC1R does a little bit. Okay, so here's a melanocyte. This is a pigmentation-producing cell. In mammals, we have 2 types of pigment. We have dark eumelanin, so this is brown to black. This is light pheomelanin, which is blonde to red. So you can look at your hair and determine which type of pigment you have. And then it's just the density of pigment that makes you brown or black or red or blonde haired, for example. MC1R really is the switch on melanocytes that determines what type of pigment you produce. And so when you turn on MC1R, levels of cyclic AMP go up and you get dark pigment. If you turn down MC1R, you get light pigment. So our hypothesis is that this mutation, this ARG-to-cis mutation at position 65, either reduces the receptor activity so that there's less cyclic AMP and we get more blonde mice, or it reduces how well these ligands bind to the receptor. And if they don't bind as well, they don't get turned on as well, and then you also get blonde mice.
Is it the same gene that is affected when you have a color change in hair?
Oh, so like Arctic hares or do you mean in humans or in?
Both.
Okay, so we actually know very little about color change. So let's say seasonal change in Arctic hares or in weasels, et cetera. So we have no idea. But remember, this is a mutation in the gene. So when we have seasonal color changes, it's hard to explain by just a single mutation in a gene. What probably is happening is you have some hormones that come in that change during the season that act through the same pathway, but may not be a genetic mutation in MC1R.
Right. Hormones also be for people, probably?
Yeah.
From little kids to older kids.
Yeah, so your hair changes with age, and then it's a little different than getting gray. So I always get asked this question, so I'll just answer it. When folks get gray, what happens is that the melanocytes just aren't being sort of regenerated. So the melanoblasts, which are sort of like the stem cells of melanocytes, just start dying, and so you don't get as much melanocytes, so you don't produce as much pigment in general. And that leads to gray hair. We actually don't know that much about that process either. So in some ways, it's easier to study genetic changes where you have two forms that are different as opposed to one form that changes over time. Okay, so what's nice about this system is that we have this mutation that differs between dark and light mice. And what we can do is actually take the receptor, express it in cell culture, and then ask if the two forms that differ by this one amino acid behave differently, and address how they behave differently. And so let me just remind you, alpha MSH turns on MC1R, and then you get, when it's turned on, you get more cyclic AMP and you get dark. So,
where does that come from?
Alpha MSH. So it's a hormone that circulates in your body. It's a byproduct of a gene called POMC that actually gets cleaved into lots of little hormones.
Okay.
It's also associated with tanning, and so you can change the amounts of hormone as well.
Okay, thank you.
Sorry, it's actually a hard question to answer. Okay, so what we can do is we can express this in cell culture and then add alpha MSH, which we can just buy, and then measure cyclic AMP as a proxy for activity. And we just want to know if the two receptor types differ. And so here we're adding more and more alpha MSH in this axis, and this you can think of as a proxy for receptor activity. And so here is our dark allele from our dark mouse. Here's the receptor activity. You add more and more of the activator, and you get this sigmoidal response curve. This is sort of the typical thing you would expect, and then it sort of plateaus out, and that's as active as the receptor is going to be. If you do the same assay with a receptor that just has that one amino acid change, from the light mouse, you can see that it's much less active, no matter how much alpha MSH you add. And this is consistent with the idea that it contributes to light coloration. So let me just pause for a second and say, here what we have is a mutation in a receptor, a single nucleotide change that codes for an amino acid change that causes a change in the receptor activity. We know that this receptor activity causes a change in pigmentation. And in these mice, we know that a change in pigmentation causes a change in survival. So we've made a link between a single nucleotide change and survival of mice in the wild. And so this is a really nice sort of example of genetics all the way up through fitness. I'm not going to go into detail. I might skip this next slide, but just to say that there are two additional genes. Agouti. So remember, MC1R is just a single 1,000 base pair simple gene. Agouti is super complicated. It's got multiple promoters, little coding exons. And in fact, with Agouti, unlike MC1R, we didn't find an amino acid change between dark and light mice. But we found a change in expression of Agouti. And so this immediately says it's more complicated. And it means that the mutations are probably somewhere in this big regulatory region upstream. And then this third gene, even more complicated, there's 22 exons. We found one coding mutation in corin, but it doesn't look like it's in a particularly important part of the receptor. But we do again see a change in expression. And so again, it's probably going to be a regulatory change. So these two genes are much more complicated in their effects. So while we sort of nailed the story for MC1R, the case for corin and Agouti are a little bit different. But if you remember back to that slide I showed you previously, Agouti actually interacts physically with MC1R, and it's a repressor of MC1R. So it makes sense that if you increase Agouti expression, that's the same thing as decreasing MC1R activity, and it leads to a lighter color mouse. Corin was a gene that was just discovered about, maybe it's 2 years ago now, by a group at Harvard Medical School. And corin seems to interact in the same pathway, at least, with Agouti. And what we find is you increase corin expression, you also get lighter coloration. So now we've sort of got the pieces of the puzzle that all sort of fit together. And then the last thing I want to just sort of tell you about before taking you from the lab and into the field is that we're also trying to understand how changes in these genes produce the change in phenotype through development. And so to do that, we do a lot of work with mouse embryos. And so I told you about MC1R. I just want to give you a little hint of what we do with these other genes. So the first question is, with agouti and corn, there are changes in expression, but these are likely to be changes in expression during development. So while these embryos are developing, and that's because these are just newborn mouse pups, you can already see the color differences when they're born. And so that means there's something happening during embryogenesis that is controlling these pigmentation patterns. And so what we're able to do is look at expression through embryogenesis. So here's a gene. So here's, now you're taking the mouse and facing it towards you and cutting this way. So you're looking through the core of the mouse, and this is the dorsal ventral boundary. And what we can do is use antibodies and probes to look at expression of these genes through time. So this is an embryo at day 12. And this particular mouse, you can see agouti is being expressed in the belly. And then we can look at where it's expressed in the skin. It's expressed in the dermis, not the epidermis. And then we can see it's expressed in particular in the dermapapilla. So this is a hair shaft where the hair is going to grow, and this is sort of the base of the hair shaft. So as that hair is growing through time, pigment gets deposited on it as it grows, and all that's sort of happening around this area, the dermapapilla. So here's based on those types of studies, what our model is. In the mainland mouse, agouti seems to be expressed in the belly. So again, you're looking at a cross-section of a mouse, so facing you, sort of cut in half. Corn has a very complementary pattern. It's expressed in the back. And we think that in beach mice, what happens is that you overexpress agouti, and then the corn expression retracts. And that changes where that dorsal ventral boundary is. And so if you remember with these beach mice, what looks like happened is instead of being white-bellied and dark on the back, sort of halfway, it just sort of gets shifted up. And we think what's causing that difference is really this interaction between corn and agouti during development that leads to these differences in color pattern. So MC1R seems to be making the mice lighter in color, but it's corn and agouti that seems to be changing the pattern, that patterning in color. Okay. So now I think we sort of have at least a hint at each of these pieces. We started with phenotype. I've showed you that natural selection seems to be driving this. I've told you about the 3 main genes that underlie these differences and given you a little hint or a taste of how these genes through development produce that variation in phenotype. And so I like to think that we start starting to form this complete picture of adaptation. And in some ways for us, this is just the first step. Because now that we have this complete picture, we can go back out into the wild and actually learn about how these traits evolved. Okay, so we actually do this. We go out in the field. This is one of our field sites near St. Augustine on the eastern coast of Florida. And we spend a lot of time out in the field trapping mice. So we do a lot of marker capture studies, for example. So here's a mouse with a little ear tag that we've caught. We take DNA samples, taking little tail tips from them, and then we also record their color variation, and then release them back in the wild. So just by way of reminder, everything I've told you about was a cross between the Santa Rosa Island beach mouse number 3 and then a dark mouse from somewhere in central Florida. But there are a number of Gulf Coast subspecies, so 5 of them. And then the Atlantic coast, there's 3. And actually number 7 has gone extinct in the last 50 years or so. So the other thing that I'm not going to talk about is that these subspecies, many of them are endangered or threatened because they live on these beautiful beach habitats where we like to build summer homes. We bring cats, which there's a big feral cat problem in this area, and then there's lighting issues. And then there's this oil spill, which is not going to be good either. So there's a lot of issues associated with the long-term survival of these mice. And this subspecies on the Atlantic coast going extinct is just an example of that.
Could you say something about how the subspecies were determined?
Yeah, so the subspecies were largely determined based on color. They were described, again, initially by a guy named Howell in 1909, and then by Sumner later on, and then they've been slightly more defined. So let me show you this next slide. So here's just a cartoon representation of each of the subspecies, and you can see that they each differ slightly in their overall color pattern. So all of the beach mouse subspecies are lighter than the mainland form, but they're pretty distinct. And so you could give me a mouse from one of the Gulf Coast populations, and I could tell you with probably, on a good day, 95% certainty which population it comes from. So these are pretty distinct. And again, this is the one that we did the crosses with.
Does the color of the sand vary in all those areas?
Yeah, so I think I took this slide out. So we've done studies, that's a great question. So why do they vary so much? So we've done a study that was just published last year that suggests that natural selection has sort of tweaked the pigmentation patterns of each of these subspecies to most closely match its habitat. There's a correlation with sand color. So for example, these guys are closer to this outlet of the Mississippi River. I'm not from the South, so I think that's what it comes out here. But it's much, you can go out on the, and I can claim jet lag. But it's much darker. The soil's got a lot more soot and debris in it, and so it's much darker. But I think it's also associated with vegetation. So for example, these Choctawhatchee beach mice are very orange. And you go out there, and it's not that the sand in some sense is a little bit more orange. There's a lot of sort of coral and shell debris. But there's also this vegetation that in the evening, the whole place glows orange. And it's sort of an anecdotal observation, but nonetheless, there seems to be a really nice correlation with their environment and their precise pigmentation pattern. Santa Rosa Island is by far the lightest and has the least amount of vegetation. Okay, so all the beach mice are lighter compared to the dark mice, but they're distinct. Yet if you look at the Atlantic coast mice, they're about, I think, 300 kilometers away. And there's some strong similarities. If you handed me these two mice, I couldn't tell you the difference. I couldn't guess. I could guess, but I wouldn't be right. Maybe 50-50. These guys are actually quite similar in color, and these guys are very similar in patterning. And so this raised the question of, first, that those genes that we found on the Gulf Coast, are they contributing to light coloration on the Atlantic coast? So the first thing we wanted to do was just make a phylogeny, a tree that showed the relationship of these subspecies. And this is sort of just a simplified version of this. So all the 5 Gulf Coast subspecies are all very closely related to each other. And with these genetic markers, we couldn't tell you which ones are more closely related. But what's really interesting is these Atlantic coast subspecies aren't actually that closely related to the Gulf Coast subspecies. In fact, they're much more closely related to dark mice from central Florida. And these Gulf Coast subspecies are much more closely related to dark mice from the panhandle of Florida. And so what this suggests or raises the possibility then that light coloration has evolved twice. So these guys probably arose from a dark ancestor and evolved light coloration. And these guys did so independently. Okay, so that's what this topology, these relationships suggest. Yet what's great now about having the genes in hand and the actual mutations that cause at least some of these guys to be light is we can ask, well, do those same mutations exist in the Atlantic coast mice? And so we sequenced MC1R. The first thing we did was we asked, is this mutation found in any of those Atlantic coast mice? And so we sequenced the part of the gene, and to our surprise, none of those Atlantic coast mice, despite the fact that they looked phenotypically indistinguishable from the Gulf Coast, had this same mutation. We even went to those extinct subspecies and were able to get DNA out of museum skins that are housed at the University of Florida Natural History Museum, and we still didn't find this mutation. So then the next question will maybe, what if it's not this mutation? What if there's another mutation in MC1R? Because I told you there's tons of mutations in MC1R that can cause light coloration. So when we sequenced the entire gene in those Atlantic coast mice, we found 4 mutations shown in yellow. But when we did those same pharmacological assays that I told you about, where we expressed them in cell culture, none of these mutations had an effect on receptor function. So what this suggests to us is that there are different genetic solutions to that common ecological problem of wanting to be a light mouse living on light soil. So MC1R doesn't seem to be involved in causing light coloration on the Atlantic coast. And currently in the lab, we're working on Agouti and Corin to ask, are there changes in expression in these two genes that may be causing them to be similar in coloration? But it could be a completely different set of genes, of course. Okay, so again, I've given you a little taste of, once you have these sort of understanding of the ultimate and proximate mechanisms of phenotypic variation, sort of what you can do out in the field to try to understand how these traits actually evolved. So I want to end by just telling you one more quick story. Because here what I've done is I've told you a story about how within a single species of these Peromyscus polynotus, you had different genes involved in producing similar phenotypes. But sometimes we have the same gene produces similar phenotypes in really different organisms. So at least to a mammologist, perhaps the most different organism from a mouse may be an elephant. I'm going to tell you a story about a relative, and that is of the mammoth. So I had some colleagues in Leipzig in Germany. They're now very involved in sequencing the complete Neanderthal genome, which I'm sure you've heard about. It's in the news. But even just 4 years ago, it was a huge deal to sequence just a single gene from an extinct organism. And they were initially working with mammoths, and in part because mammoths are sort of the perfect extinct organism in the sense that their remains have been sort of frozen in permafrost for about 14,000 years. So they're not quite as old as Neanderthals. And their DNA has essentially been in this giant natural freezer, so the best type of condition to preserve DNA. So when they started this, they had sequenced some mitochondrial DNA, but that's relatively easy to do. I don't know if you guys have talked about that, but nuclear genes are a little bit more challenging, and so they wanted to pick the simplest nuclear gene. Well, MC1R, I told you guys, it's a pretty simple gene. It's only 1,000 base pairs. There's no introns. It's very straightforward. So they decided to sequence MC1R. And what they found when they sequenced MC1R in, I think it was 5 individuals from a single site, was that there was a mutation that was polymorphic. Well, it turns out that mutation that was variable was the exact same mutation we found in our beach mice, an arginine to cysteine change at position 65. So that raises the possibility then that mammoths like our beach mice may have been variable in color. Now these were DNA samples, unfortunately, taken from bones. We don't know what the actual hair color is, but there have been samples of hair that have been recovered, and they do seem to vary from more reddish to more brown. So we published these, our beach mouse paper in this paper in the same issue. And of course, the press picked up on the mammoth story like crazy, so the headlines read things, like blonde mammoths have more fun, et cetera, et cetera. There are also a number of other species, which I was sort of hinting at earlier, that vary in coloration due to mutations in MC1R. I'm not going to tell you, I think just for the sake of time, all the details of this study, but it's work I've been doing with Erica Rosenblum at the University of Idaho, looking at lizards that live in White Sands, New Mexico. So now we're traveling from Florida to central New Mexico. You can see from the satellite image that there's White Sands. It's a giant sand dune. It's right near the White Sands Missile Range. I don't know if you've been there, but it's a really neat area. And that's what the White Sands look like. So it's like walking on the moon. And that's what the surrounding habitat looks like. And in these areas, you have lizards that are pale on the sand dunes, and this is sort of the regular phenotype, this particular species of lesser earless lizard. What's neat is it's not just this species, but it's the eastern fence lizard and the little striped whiptail as well. So all of them have converged. And we found mutations in MC1R, different mutations, but each one of these comparisons has a difference that segregates perfectly with, or nearly perfectly with color. And so we're able to do those same pharmacological assays in each of these and found differences in some of them associated with changes in color. I'm going to skip these slides because they're a little detailed. But let me just leave you with sort of this idea that finding the genes underlying color differences really allow us to say something about convergence at the genetic level. Do the same genes contribute to differences in color? So of course, there are many examples where there are different genes involved in similar coloration. So I told you about the Gulf and Atlantic coasts. MC1R is involved in the Gulf coast and not the Atlantic coast. In some cases, in very divergent species, you can have even the same mutation associate in the same gene associated with similar color differences. In mammoths, I'm waving my hands a little because we don't know for sure about the color differences. In the case of mice and lizards, sometimes there are different mutations in the same, in this case, MC1R, that lead to similar phenotypes. And then I didn't tell you the details about this, but sometimes you have different mutations in the same gene that cause the gene to do something different, but again, to lead to the same phenotype. So I've hoped to give you some examples of how understanding the genes that are involved can tell us something about the evolutionary history and about convergent evolution at the genetic level as well. And MC1R, as I told you, is involved in a number of different examples of color variation. It's not just camouflage, though. It's also involved in sexual selection, so assortative mating, where, let's say in this case of these Arctic skuas, dark ones prefer to mate with dark birds, and light ones prefer to mate with light ones, and these color differences are associated with MC1R. That's similar with snow geese and banana quits and manakins. And so this raises this interesting question of why MC1R? Why does MC1R seem to be involved in lizards and mammoths and beach mice and pocket mice and all these birds that I showed you? I think there's two explanations for this. One I think is biologically uninteresting, and then there are several that are more interesting. I think, as I told you with MC1R, it's such a simple gene and it's so well conserved, you can sequence it in any species, right? So you can sequence it in mammoths. They didn't necessarily think to sequence MC1R in mammoths because they thought there was a color difference. It's just an easy gene to look at. But I actually think that there's a more interesting reason. And this has to do with, sorry, I've used this sort of jargon, this idea of minimal pleiotropy. And what that means is that MC1R really only affects pigmentation. I just came back from a meeting in Holland, which was 2 weeks ago, that it was called the melanocortin meetings, and so this is quite a specialized meeting, but nonetheless, there's a lot of people sort of trying to understand what the effects of mutations in MC1R and other melanocortin receptors are, and if they affect other things. So this is still sort of a hotbed of controversy. Nonetheless, it seems like at least the predominant effect is on pigmentation. So a lot of, remember those other genes I was telling you about in that list of candidate genes, you change color, but you also change body size, or you change, you have other deformities or other things. With MC1R, it really seems to be associated with pigmentation. So you can change this gene, you can futz this gene and really just affect primarily pigmentation. Also, it has large phenotypic effects. A single amino acid mutation can take a lizard that was brown and make it white. So it doesn't require a lot of changes. And then the third reason is I think it's a pretty large mutational target. So I showed you that image, and I'll end with this image. Here are all the mutations in MC1R that are associated with changes in natural populations. So adaptation to either adaptation in terms of camouflage or sexual selection in terms of those examples of birds I gave you. So it's not that you have to wait around for a single mutation in this gene. You have lots of different potential changes in this gene that seem to affect pigmentation. So I think this is why potentially we see MC1R as being sort of a hotbed for evolutionary change. Okay, my last slide, just to say that I think that we're at this really exciting time in evolutionary biology, because a lot of those questions that motivated Darwin, we can now start to answer using molecular tools. So sort of inspired by Darwin's interest in natural history and his grounding in fieldwork, yet armed with molecular techniques, we're sort of starting to unravel what he referred to as that perfection of structure and co-adaptation, which most justly excites our imagination. That is the genetics of adaptation. And that's my lab group. This is a picture taken outside looking our mousea. So a lot of these folks contributed to the work that I told you about today. And with that, I'm happy to answer any questions that you guys may have.
Could you go back 2 slides to that?
Yep.
Yeah. So is it these mutations that lead to conformational change that you think is leading to the receptor being more or less activated?
Yeah, so because this is a transmembrane domain, it's really hard to get structure because once you take it out of the membrane, then it loses its conformation. So how much any of these mutations change structure, we can model that, but it's really hard to experimentally test. What we do know is that at least in some of the cases where we can do those sort of pharmacological assays, that mutations will either, so for example in those beach mice, I showed you that this mutation here affects receptor signaling, so you don't ever get as much cyclic AMP. I didn't show you was another experiment we did which showed that the reason that it doesn't signal as well is that it doesn't bind alpha MSH as well, and that's what activates the receptor. But it's kind of weird. I mean, this is internal to the cell, and alpha MSH binds externally. So you sort of have to intuit that there's some sort of conformational change that affects that. Now with the lizards, the slides that I flipped through, we found 2 interesting things in terms of the receptor. One was a signaling effect, like beach mice, but another mutation in this receptor caused it to not integrate as efficiently into the melanocyte. So you had fewer receptors, and so that also had the effect of reducing signaling, but through a sort of fundamentally different functional change. So maybe why there's so many types of mutations that can affect coloration is because there's lots of different ways to either change signaling, like either affecting ligand binding or just reducing signaling or reducing its efficiency in terms of integrating into the membrane. So I think that gives it a lot of flexibility and contributes to this.
How similar are the genes when we're talking about them, like between the woolly mammoth or even the lizards and here? How much alike are they?
So in terms of MC1R, it's really highly conserved. So the structure is always the same. One thing that does change a lot is this first extracellular region here, that sort of floppy bit. It used to be thought that that sort of helped ligand binding, but in fact we know you can chop that whole thing off and it still works just fine. But what that means is that it's because it's not functionally necessary, that's the region of the receptor that changes the most. So if you took woolly mammoth and lizards and beach mice, that's where you see a lot of change, both in terms of amino acid changes, but also in terms of length. So some lizards, for example, are 5 or 6 amino acids shorter than, let's say, beach mice, but that doesn't seem to really have a large effect. In terms of the rest of the receptor, it's pretty well conserved. I mean, you can take primers and put, if you're doing a PCR, primers in each end of the receptor and amplify it in almost any vertebrate.
And in all of these organisms, well, I assume MCR then must be a mammalian characteristic, and in other?
Across
is it
vertebrates.
Always a pigment?
Yes, in vertebrates.
Okay.
Yeah. But then there are things like MC2R, MC3R, MC4R, which are part of a larger gene family. And MC4R, for example, is expressed in the brain, and mutations in MC4R are associated, one of the few, I learned this last week, one of the few genes that mutations in this genes are associated with obesity in human populations. So remember I told you about agouti being overexpressed and you get these plump mice? That's because agouti interacts, instead of with or in addition to MC1R to cause the color change, also interacts with MC4R in the brain, which is associated with refeeding behavior. And so that's why you get both pigment change and the obesity phenotype. But this is a very, very well conserved gene, which is, again, why I think it's so well studied in some sense, especially in natural populations where you maybe can't bring everything in, or like our lizards, you can't bring them into the lab and breed them so easily. And so we just thought, well, let's give it a shot and go for MC1R, and in some sense got lucky, or yeah.
Back to white sands and the lizards. At the intersection between those two habitats, what phenotypes do you see? And are there behaviors that keep the white lizards where they're safe and vice versa?
Yeah, okay. That's a great question. So Erica, who is at the University of Idaho, has just got back from the field, and they're just showing some really interesting patterns in terms of mate choice. Okay. So first let me say on the ecotone, so on that edge, you get sometimes lizards of both types. And it depends. So you do get lizards of both types, and presumably the dark ones that went to the light soil probably get picked off more often, and the light ones that dribble off. But as you move further away from that ecotone, you find fewer and fewer mismatched lizards, and that's probably just selection. They're not getting that far on the wrong direction. And again, selection. They haven't quantified selection. They're starting to build a little plasticine lizards to do and actually it should work better because lizards are diurnal, they're predators. But the problem is the plasticine melts in the day, and so they've got, the lizards lose their shape. But I'm sure they'll overcome that challenge. But so I don't think they have an actual estimate of selection yet. But one benefit about working on lizards is that color matters also in terms of mate choice. So it's more of you can think of it as a trade-off between natural and sexual selection. So these lizards, let's see if I can get back to these quickly, also have these really bright patches, which you can't see because these are pictures of the dorsal side, but eastern fence lizards have these bright blue patches on their sides, and either for territorial defense or sometimes for attracting females, they do these push-ups. I don't know if you've seen these. It's really, you can see them on YouTube. They have these little videos, right? And they flash those blue patches. And these guys are, again, they have blue heads and they have really bright blue tails. You can see it sort of muted out here. But when you turn pale, you're more camouflaged, but you also mute those sexual signals. So there's that balance between survival and then attracting a mate. And what Erica's group has recently shown is that there's some evidence for mate choice. And that is, let's see if I get this right. I'm pretty sure it's assortative mating where these guys tend to prefer light guys and these guys tend to prefer dark guys. But these males will sometimes these females will sometimes prefer these more than these. And that has to do with the coloration. So the females are choosing, will choose the bright colored males of these guys, but these females don't like the dull ones of these. And I don't remember what the ratio is in terms of the pale ones, if they prefer pale ones over the bright ones of these males, but the other direction, it's always true. So anyway, there's a complicated twist in this story, which we don't see in the mice, and that is that color matters also for sexual selection.
Would that be maybe the females are choosing the ones that can survive even in a bad environment? Does that mean you're stronger, I suppose?
Well, it's hard to know how they would know who would survive better. So I think it's hard to imagine how that could be the case per se. But it's sort of, it's not like these guys, let's say they prefer light colored guys more. It's not because they sort of know that their offspring are going to be light colored and therefore they should, date and marry a light colored lizard. But it's sort of, it gets the ones that do have variation in choice and the ones that do prefer the light ones and have light offspring, then those genes get passed down preferentially, and so it sort of gets co-opted in that sense. So it happens, but in a slightly less deterministic way. Yeah.
Did I understand that you gave the mice the alpha MSH and then it increased the melanocyte production of the darker pigment?
We give the cells that have, so the way we do these assays is that you take MC1R and you put it in a vector, an expression vector. So it's got a promoter, so it'll express MC1R. And so you have the beach mouse MC1R and this manufactured expression vector. We put them in human embryonic kidney cells. These are just a cell line that's very easy to maintain. They're sort of starter cells. The easiest ones to maintain in the lab. And then we give them mouse alpha MSH. So you just put that in with the cells. So it's like circulating levels of alpha MSH. And then we measure cyclic AMP. So this is all done in cell culture.
So we don't have like a tanning pill here?
No, there are people that are really interested in tanning and, of course, the links with skin cancer and all of this. So there's a big group actually here in the dermatology department that we meet with once in a while because we obviously have very different perspectives, but we both think about the same pathway. But what would be nice, I mean, what we'd like to do with the mice is to actually use transgenics to see if we could recreate a light-colored beach mouse just by changing Agouti core and an MC1R. And that's sort of the gold standard, you know, the ultimate proof. But it's also a lot of work. So these cell culture assays are a nice way to show that the receptor is different, and it's different in the direction you expect if it's going to lead to light coloration. And it doesn't cost quite as much, and it's not as much of a time investment and so forth.
I also heard, I don't know if this is true or not, maybe you can validate. I heard a rumor that Darwin actually had, like on his bedside or something, a version of Gregor Mendel's work in German that was untranslated or something along those lines. Do you know about that?
Yeah, so so there's somebody on campus, Janet Brown, who's the world's leading expert on all things Darwin. Okay. So she's the right person to ask directly. What she has told, my understanding is that there wasn't evidence that he owned a copy or that he read a copy. And so the general sense, I think, is that he actually didn't read Mendel. Maybe that he had a copy but didn't read it, or, but certainly even, you know, Mendel was sort of rediscovered. So even though Mendel, you know, those papers came out early on, they certainly weren't appreciated until much later. My understanding, yeah, there wasn't a direct link, but Janet would be the one to, yeah, maybe I'll email you back so I get you the sort of right story about this. Janet knows for sure.
I just read in Sean Carroll's book this morning that, on the subway in actually, that Mendel's paper was available in England, but there was no evidence that Darwin had a copy of it.
Yeah, but there's been some back and forth about this, but I think that's my understanding too, is that there's no direct link, even though it was available at the time. But it was available to lots of people who, yeah, just at the time didn't appreciate it, which is kind of, you know, is very interesting in and of itself.
You mentioned early on when you were showing the different skins pinned out that you measured the coat color. How do you do that? Do you quantify that?
Yeah, so that's a great question. That actually turned out to be much harder than, you know, we sort of thought of in the beginning, you know. So we do it in a number of ways. Quantifying pattern actually turned out to be a challenge because you can imagine if you just measured the area of the coat that was pigmented, you could get the same value for a mouse that had pigment on its nose and not on its tail as one that had pigment on its tail and not on its nose, or vice versa. And so what we decided to do initially was to pick 9 areas of the body that varied the most between mainland and beach mice. So the tail stripe, the width of pigmentation on a sort of flattened mouse, the nose changes, cheek patch, they get these little eyebrows, things like that. And then we would measure them in 2 different ways. One, we just measure the amount of pigment on individual hairs. And then the other way we measured it is with a spectrophotometer, which gives us a measure of reflectance. That's completely, you know, so objective. And the reason we do both is because the way that hairs lay down on the coat of a mouse are sort of like this. So here's the tip. And really the tip is the only thing that's visible. And so you could have a mouse that had dark tips and had pigment at the base, and mice that had dark tips and didn't have pigment at the base. And the spectrophotometer would tell us that it's the same reflectance because what's visible is just the tips. But to, you know, a geneticist, those are fundamentally different hairs because one has pigment on the base and one doesn't. And then, of course, the spectrophotometer in some sense is more relevant to a predator because all they're seeing are the tips. But again, to a geneticist, they're different. And so the point being, we measured it in lots of different ways, trying to take all the information into account. And one thing that's becoming very apparent, I think, in evolutionary genetics, or these types of studies that are trying to link genotype with phenotype, is the genetics is getting really easy. We can sequence genomes, and finding all the different variation in a genome is not that hard. Measuring phenotype is much more difficult. It's an unbounded space. And so coming up with ways to accurately characterize phenotype and to do so sort of objectively is actually hard. And pigmentation may be one of the easiest traits to measure, really. I mean, it doesn't change really over the lifetime. You can keep these museum skins in a cabinet and go back and measure them multiple times. We have some projects where we're trying to find genes underlying behavior, and it turns out that that's a lot harder. There's a lot more environmental effects, and how to measure behavior is a huge challenge.
So in your field studies, you said you would take DNA samples from the tails. Do you take fur samples, or do you have to kill the mouse to get that?
No, what we do is we do these measures of the 9 spots. We have this sort of spreadsheet, and we fill it in, and we try to get the same person taking all the measurements. So we actually get the sort of simplest phenotypes that way. So we can get a lot more information from the study skins than we can from a live mouse, but then, releasing the mice, and we can get a lot of information about natural selection. And so we're doing studies, for example, to ask if mate choice is important. So we have the phenotype of the mouse, and then we can genotype all the offspring and see who they mated with. And we're finding some evidence for habitat selection. That is, light mice tend to live in patches that have lightest soil and least amount of vegetation, and darker mice. So these types of field studies are actually really important, but we do lose a lot of information about phenotype.
Is there an organism that's in New England that have similar studies being done on?
That's a great question. There are some old studies of pale mice living out on some islands off the Massachusetts coast, and I can't remember what islands they are. This is a study, published I think in the 30s or 40s, and so that's one thing that's sort of on our list to go do, trap some mice and see if there really are color differences and how big those are. We trap locally. We've been doing some work more recently with chipmunks around here, so it's related in the sense that we're interested in how patterns are formed. So we want to know how chipmunks get their stripes, and so we go out sort of locally and trap chipmunks, but it's a little bit different from this. And the why chipmunks have stripes is a little bit less clear. There are, I don't think there's any. I mean, in the Southwest, there's lava flows or these those really extreme habitats, but I can't think of anything local. But I'm also relatively new to the East Coast. So.
Thank you.